So I guess it is time. And as I said, it's my privilege and honor to introduce to you Rudolf Jaenisch from MIT. And I guess you have to tell us the title of your talk because I don't have it with me.
Okay.
Rudolf, please.
I will do. So let's see if I get this here. So my title, sorry, I missed to send it. So the title of it will talk about Rett Syndrome and really the biology and therapeutic approaches. And why doesn't I can't? Oh, and just to in disclosure, I'm advisor to a number of companies: Fate, Fulcrum, Omega, Camphor, and DuPont. So Rett syndrome is a very serious disease. It's caused by mutations of the MECP2 and methyl binding domain protein, which is on the X chromosome. It affects only females. Boys die early in general. The onset is 1 year of age and is very severe mental retardation, autism, no speech. It has these very typical severe hand movements, this clutching hand movement, other movement abnormalities, and they're microcephalic. Now, most mutant mouse have been generated in Adrian Bird's laboratory and my laboratory more than 20 years ago. And these most mutants really resemble the human disease very well. For example, if you lift a mouse at the tail, it spreads its paws. If you lift a mutant mouse, it clutches. And this has many other very similar phenotypes as the human. So why is Rett syndrome, I think, an exceptional therapeutic target? It is a severe disease. It is not rare, 1 in 10,000. It has a well-defined genetics. And the mouse mutant is a faithful model of the human condition, as I said. Similar molecular and functional phenotype, in vivo and vitro, relevant readouts for any manipulation you want to do. But most importantly, this disease is not developmentally set, but the most serious symptoms can be reversed postnatally. I will come to this. That makes it, I think, an exceptionally attractive target for therapy. So MECP2 binds to methylated CpGs. It recruits these co-repressors and core and SMART, and really preferential binds to methyl CpG, but also to methylated CPA dinucleotides. And it has several domains: methyl binding domain, and then this long domain, the repressor domain with the NID, which recruits NCOR and SMART, and other domains we'll come to later. So the phenotype of Rett neurons in human, in vivo and in vitro, is the mutants are smaller, they have shorter dendrites, and they're electrically less active. Gene expression changes, and MECP2 inhibits gene expression. Adrian Bird showed this 30 years ago, but MECP2 also acts an activator of gene expression. We and others showed that. So that's a bit confusing. So as I said, mutant neurons are smaller than wild-type neurons in females, and they have less complex dendrites. And when you plate neurons in a multi-electrode chamber, electrode array chamber, then what you find is that wild-type neurons really, over 2 months or so, really become spontaneously very active. Mutant neurons, really almost no activity. There's a very strong phenotype in culture. So the Rett mutant neurons show smaller size, cell size, so reduced complexity of neurites, and reduced electric activity. So wild-type neurons have a certain size, certain RNA and protein concentration. Mutant neurons are smaller, have less RNA and less protein. But you can reverse that. And you can reverse it by several ways. So first of all, I want to say that we believe that Rett is likely a very unspecific but metabolic disease. It affects the gene expression of ribosomal genes and of mitochondrial genes and others. But you can reverse the phenotype by either re-expression of MECP2 in mutant mice that rescues the lethality, or by molecules like BDNF, IGF-1, and KCC2, which is an ion transporter, who really delays disease progression in mutant mice and restores the mutant phenotype in human neurons. So this is really the useful readout for any manipulation. So there have been different therapeutic approaches to Rett. One is just gene transfer, transferring this MECP2 into into mutant neurons in mice, for example, you will get defined MSP2 expression. But the challenge is that you very likely get overexpression by viral transduction. And overexpression of MSP2 is as toxic as no expression. So that's a challenge. Some people try to reactivate the inactive X where the mutant, the wild type allele of MSP2 might be. The problem here is it would restore wild type levels, but two active X's have toxic side effects like cancer. People have used epigenetic drugs to activate the inactive allele, small molecule approach, but again, it's unspecific and side effects are likely. Epigenetic editing to specifically activate MSP2, this would restore a wild type level just of the gene, but again, you worry here about, you have to, the bar is here, transduction efficiency of all these editing tools. And finally, activation of other pathways, and I mentioned IGF-1, KCC2, and this really works, but again, it's not terribly efficient in mice. So I want to briefly summarize what we're doing in epigenetic editing to activate the mutant MSP2, the wild type MSP2. So just set the stage. If you have a normal individual, then every neuron expresses MSP2. But if you have a Rett phenotype, then of course only half of the cells expressed, depending on axon activation, express a wild type, and the other one the mutant allele. But of course, every cell carries a wild type allele, either on the inactive or the active X. So if you could reactivate MSP2 on the inactive X, then you would restore and rescue Rett. Now, most Rett mutations are loss of function. So it doesn't matter whether you target the mutant or the wild type allele, because the mutant activation, allele activation would be of no consequence. So the reactivation of MSP2 wild type allele and mutant allele on the XI should restore normal expression levels, not overexpression, not underexpression. So we try to do this by using the dCas9 epigenetic editor, and we used a reporter line where one allele of MSP2 was GFP labeled, the other one tomato. And then after inactivation, they were single colored, only tomato in this case. So we, by Cas9 TET fusion, we would demethylate the promoter and screen for double positive cells. And it works very well. You start with a single colored cell and you end up with double colored cells. And this reactivation restores the Rett phenotype in cultured neurons to cultured human neurons to that of wild type neurons. So if I summarize this, the demethylation of the promoter reactivates MSP2 on the XI in dividing cells and rescues abnormal electrophysiological activity in neurons when you differentiate those two neurons. However, when you try to do this in post-mitotic neurons, then the reactivation of MSP2 is really only transient, and we not quite understand this. So this is ongoing research, and we haven't really solved that yet. So the Rett syndrome really causes challenges. I think we do not understand the normal function of MSP2. Just to recapitulate, it's a proposed function from Adrian Bird. It's a repressor gene, and it is also an activator of genes, of thousands of genes, activator of thousands of genes. So the question is, are we missing a key function? And one important part is there are 14 million copies of the protein per neuron. It's at the same level as histones. So could MSP2 act as a scaffold in transcriptional condensate? That's one of the questions, and that's why I'm talking at this meeting. And could this explain the altered metabolic state of Rett neurons? And so is MSP2 protein found in molecular condensates? And this is now a very close collaboration with Rick Young's group at the Whitehead, which I'm going to talk about. So MSP2 has two intrinsic disordered regions, one at the amino end, one at the carboxy end. Mutations are in the methyl binding domain and in the IDR2, which cause Rett. So in vitro, the protein forms condensates, depending on the concentration of protein you use, and it's sodium chloride dependent. So with higher concentration, the size of the droplets goes down, and you can make this Feist diagram. In addition, in vitro, these droplets fuse, as you can see here, and they exchange rapidly with the environment. If you, after FRAP, they recover very rapidly. So the effect of DNA on droplet formation was tested. And so if you use an MCP2 concentration which doesn't give you any droplets and add DNA, you find there are droplets forming. But if you add methylated DNA, it's much more efficient, but it's the same concentration. You can see this quantified here. So of course, MECP2 is a methyl binding protein. So the DNA stimulates formation of MECP2 droplets, but methylated DNA is more efficient than unmethylated DNA. The functional regions, as I said before, there's IDR1, the DNA binding domain, and the IDR2. You can ask the question, what do these domains do in vitro? So the full length makes, of course, droplets. If you delete the IDR1, there's no effect on droplet formation. If you delete IDR2, it abolishes it. If you just use IDR1, there's no droplet formation. If you just use IDR2, there is droplet formation, although there's less smaller droplets than if you use the full length. So the conclusion would be both the MBD and the IDR2 are required for transcriptional content formation in vitro. So clues to the functional regions of MECP2 comes from the mutations. So this will be a spectrum of a number of mutations in the MBD and the IDR2, which cause Rett syndrome. And so, as I said before, the deletion of IDR2 abolishes it. Now the question is, does it occur in vivo? And in vivo, you can see here, when you really transfect these constructs into cells, then IDR2 deletion transfection really abolishes the droplet formation, but IDR1 deletion does not. So IDR2 is essential for content formation in vivo. Now, of course, we want to tag the endogenous gene, not getting overexpression. We tag MECP2 and HB1 alpha. And you can see here both of those, MECP2 and HB1 alpha, form droplets. And they're co-localized. And you can see here, they co-localize when you stain different ones. And also you can FRAP these, you can inactivate these droplets, and they recover after FRAP at a rapid rate. It's the same rate as HB1 alpha would recover after bleaching. So the conclusions would be that MECP2 and HB1 alpha co-localize in heterochromatic condensates. Both show dynamic liquid-liquid properties with similar recovery rates after FRAP. So the interaction of MECP2 with HB1 alpha, MBD1, and BRD4 we measured. And so this was again mixing MECP2 with other proteins, which are labeled differently. And you can see here, when you mix it with HB1 alpha, they co-localize. But if you mix it with MBD1 or MET1 or with BRD4, they do not, they exclude each other. And you can see it's quantified here. In vivo, it's the same thing. MECP2, as I said before, HB1 alpha co-localize, but they're very different from droplets formed by MET1. So MECP2 associates with HB1 alpha, but not with MET1 or BRD4. So MECP2, as I said before, in neurons, it's very highly expressed, 14 million copies per neuron. So we wanted to look this in mice, in brain of mice. So we generated a reporter mice, which have the MECP2 tagged. And you can see here in these neurons, in the brain of mice, they find these big droplets, which are very different from droplets with other mice from RBP or MET1 tagged genes. So MECP2 forms transcriptional condensates in the brain of reporter mice, and we asked the question, in these brain nuclei, could you, if you laser inactivate it, would they recover? They recover with the same rate as we've seen in cultured cells. The conclusions from this all is MECP2 forms highly dynamic heterochromatic condensates in mature neurons. And MECP2 condensates in neurons are much larger than in ES cells, probably correlating to the higher expression. And MET1 forms smaller transcriptional condensates, which are mutually exclusive of MECP2. So the idea would be then both of those of the region of MCP2, the DNA and the methyl binding domain and the IDR2 come together to form this condensates. And so the question is condensates affected by mutations causing Rett? And there would be either mutations in the DNA binding domain or in the IDR2. So these are the mutations we looked at, they're in red. And when you, for example, take the one in the methyl binding domain, these here, then they do affect condensate formation. So R133C, this in here, and T158. So mutations in MBD affect condensate formation. How about in the IDR2? So we use these other mutations here, and you can see again, the mutations, either this one will talk about more about R168, it's truncating mutations, it's the beginning of IDR2, and these other as point mutations, some of those, they do affect condensate formation in vitro. So the conclusions, both Rett mutations, the MBD and the IDR2 affect condensate formation. So there was an interesting paper from Adrian Bird, where he used a truncated MECP2 to rescue partially Rett mice. And so it's a mini gene, which really, if this is all the constructs he had, I'm talking about this mini gene which only has the N-core binding domain, but all the rest is deleted. And that rescues mice partially. So is it sufficient to drive condensate formation? So we made this construct, and you can see as the mini gene perfectly forms condensates. And also the mini gene is able to recruit HB1 alpha to the droplets. So NID is sufficient to recruit heterochromatin factors such as HB1 alpha. So this truncating mutation 168X is a very frequent mutations in the disease. Really, how does it affect condensate formation in vivo? So we tagged it in cells. You can see here the truncating mutation really eliminates condensate formation. And also, when you look for co-localization with HB1 alpha, it's highly diminished. So then we asked the question, what about gene expression in those cells which express the truncating mutation, either in ES cells or in neurons derived from those? And what you can see here is that really about 8,000 genes are reduced with the truncating mutations if few are activated. So in a way, this is a bit counterintuitive because we are interfering with a repressive factor and you get diminished gene expression, reduced gene expression. So we believe this is not a direct effect, it's an indirect effect. We don't quite understand the mechanism. Maybe it's by redistribution of other factors when you eliminate the function of MECP2. So the conclusion would be this mutation, this truncating mutation, reduces partition into heterochromatic condensates and reduces expression of 8,000 genes. So this is all what I wanted to say. And let me just summarize the Rett syndrome and these condensates. Is this the missing link for explaining the role of MECP2 in the disease? Clearly, we have to test this in human neurons and in mutant mice. But I think of interest to us is, can we affect MECP2 condensates? Could this be a novel therapeutic application to alter the metabolic alteration of mutant neurons by targeting condensates? So that is an open question of great interest to us. And of course, is it relevant for other diseases? And so we're quite interested in that. So then we come to an end here and just acknowledge the people who did this. It was in my laboratory, Sean Liu with other people in the lab contributed, and a very close collaboration with Rick's group, particularly with Charlie Lee and Elliot Coffey, and quite a number of other people. So thank you very much for your attention.
Thank you, Rudolf. Can I ask a question? So what happens in the other cell of the body, not the neuron? Do you see no condensates also for in other cells of the body, and how does it change the gene expression there? Because so, MECP2 is everywhere, right?
Yeah, so the major phenotype of Rett is in the brain. But there's clearly, when people look carefully, there's a peripheral phenotype in liver, in heart, and some other. So clearly it affects it, but there's lower expression. We have not looked at condensates directly in those tissues, but in yes cells, which is much lower expression, there are condensates, but they're somewhat smaller.
Okay, could you find the SUV? What are the other partners in the condensate there?
So HBM1 alpha is the one we know.
Yeah.
HBM1 is another one which is recruited, and many others we don't know. We are just really trying to find out what are the partners which associate with MBCP2 in these heterochromatic condensates. I think that's a very key question.
So thank you very much, Rudolf, for an excellent talk. And we will move to the second speaker, who is Eran Perlson from Tel-Aviv University. And he's going to tell us about axonal TDP43 condensates drive neuromuscular junction disruption via local protein synthesis inhibition. Please, Eran.
Okay, so can you hear me now?
Yep.
Okay, so thank you very much, Yehudit. Thank you, thanks the organizers. It's a great pleasure to share with you our stories. So in our lab, we are very much interested to understand molecular mechanisms of motor neuron survival, synapse maintenance in ALS. We are talking about or studying a molecular communication mechanism within the neuron difference compartments, communication between the neuron soma, axon, and synapse, as well as communication between the motor neuron and its diverse microenvironment, with initially focusing now on the communication between the motor neurons and its target muscles that create the specialized synapse, the neuromuscular junction. Neurons are highly polarized cells. They have a very long axons that can reach to more than 1 meter long in adult human. And in order to survive and maintain their function, they mainly depend or need to know how to react to alteration of both internal or external metabolic or change or signaling. And this is largely occur via regulating a subcellular proteome by either axonal transport process or local protein synthesis. And this is what we are doing and trying to understand the mechanism. So to study the molecular mechanism of studying neuronal compartments, we set up in the lab a unique microfluidic compartment in which we grow in one side of the microfluidic motor neurons that can grow their long axons into microgroove, and on the other hand, we can grow muscle cells. So basically, we generate a simplified model that can be either genetic or pharmacological manipulated at a subcellular level to study mechanism of NMJ formation, maintenance, and disruptions. And we are running this system routinely using primary culture of mice, and now the system is being adopted also to human iPS-derived motor neurons and muscles. So with a collaboration with Michael Ward from the NIH, we got some iPS that we can differentiate into motor neurons and grow them in these chambers. We can also differentiate muscles on the distal side with the collaboration with Pim Frenkel from Rotterdam. And then using this system, we can ask several questions. One of the questions that we can ask is mechanism that can regulate axon growth and muscle innervation. So you can see in this movie the growth of the motor neurons from the soma side, which can represent, for example, the spinal cord, that sending their long axons to the periphery where it reach the muscles. And using this system, because it's a microfluidic system, we can do different genetic or pharmacological manipulations specifically at the soma, the axons, or the synapse. You can see here, for example, another movie showing what's happened when the axon reach the muscles and starts to innervate and create the neuromuscular junction. We can also use this system to study mechanisms of neurodegeneration and axon degeneration, neuromuscular junction disruption. So it's again trying to do genetic or pharmacological manipulation, trying to understand these types of mechanisms. For example, we found that when we apply oxidative stress at the distal site, at the NMJ, the synapse site, it can lead to toxicity and axon degeneration. However, when we apply exactly same concentration of oxidative stress to the soma site, to the cell body site, we saw that the cell is much more resistant, suggesting that the synapse compartment is much more sensitive to stress. Using this system, we can also track the movement, the transport of different molecules, factors, organelles along the axons, moving from the cell body compartment to the synapse, or from the synapse and even from the muscle to the axons and the cell body. We can track the movement of trophic factors like BDNF, organelles like mitochondria, lysosomes. We can track mRNA and microRNA, and also RNA binding proteins that can shuttle them along the axons. And finally, we can use this system to study specifically the structure and biology of the neuromuscular junction. We can look on calcium imaging, we can look on the muscle contractions, and use this simplified motor unit system to study different mechanisms that can regulate and maintain their function. So what we bring to the table is a unique compartmental system that can help us to study mechanism of neuromuscular junction formation, maintenance, and disruption. And as I said in the beginning, our disease model is ALS. ALS, we heard yesterday from Eran and Reuchalgi. So I will just want to emphasize that it's a fatal neurodegenerative disease. There is no drug, no treatments. And maybe like many other neurodegenerative disease, the synapse seems to be the first compartments to fall. And in case of lower motor neurons, in case of the synapse of the lower motor neurons, it's the neuromuscular junction, the meeting point between the axons and the muscle. Another thing that I want to emphasize is that in most ALS cases, in about 95% of all ALS patients, there is a mislocalization of TDP43 and RNA binding proteins that is usually or largely found in the nucleus and then is going out from the nucleus to the cytoplasm of motor neurons and to create condensate. Recently, there was a really nice back-to-back paper from Zevi Melabed from the Cleveland lab together with Klim from Kevin Higgin lab that demonstrate loss of function of this mislocalization to the TDP. However, TDP condensate is thought to have also a role, a gain of toxic role in ALS. And what we are trying to understand and what I will share with you today is how this mislocalization of TDP can regulate or contribute to the NMJ disruption that is seen in ALS. So this is what we want to understand. And the study was performed by really talented 2 PhD students, Ariel and Topaz, that I will show you in a minute, that besides singing and eating, can also work very hard and show really fantastic, amazing discoveries. So as I said, it was shown before by many groups that in most ALS cases, there is mislocalization of the TDP43 from the nucleus to the cytoplasm, the cell body of the neurons. And we wanted to understand how this mislocalization can contribute to NMJ function disruptions that occur very early in the disease. So what we did was first collaborating with Amir Dori from Shiba Hospital Center. We looked on intramuscular nerve from ALS patients or control, and we saw that there is a significant accumulation of TDP43 in the end of the axons in the muscles. Okay, so TDP mislocalized not only to the periphery of the nucleus or the cell body, but it can also reach to the end of the axons and to the intramuscular nerve. And that was, so it's not 0-1. I must say that there are also reports that there are small levels of TDP in the axons, and probably TDP serving as the shuttling of RNA in the axons, but there is an increase of the levels of TDP. And in order to understand what is the mechanism of how TDP accumulate in these axons, or how it contributes to NMJ disruption, we decided to utilize a genetic transgenic mice model developed by Virginia Lee, which is express human TDP lacking the nuclear localization signal through a doxycycline TET-off system and under neuronal promoter, which is mean that we grow the neurons all the time with dogs, and then is normal, he express only the endogenous TDP level, the mice TDP level. However, when we stop giving the mice or primary culture the dogs, then a humanized TDP is starting to express missing NLS and go to the axons. You can see here static nerve axon plans and form TDP condensate at the neuromuscular junction. So this model can mimic what we see also in the human patients. We can see this mislocalization of the TDP to the axon, and we can regulate and control it by adding or missing the dog's treatment. So looking on primary culture from these mice with and without the dogs, we saw that TDP can accumulate in point time condensate along the axons, and these axons can co-localize with the G3BP1 stress granule markers and with RNA dyes, suggesting that there are RNP granules with TDP that are forming along the axons. Next, we thought that maybe, and it was shown also that TDP can regulate translation. So we thought maybe this condensate can regulate protein translation, or maybe can inhibit translation, which is needed and necessary to stress axons like in the ALS. And to look for locally synthesized protein, we use the OPP system, which is basically a drug that can stop protein synthesis and tag to it, and then we can tag it with a fluorophore, and then we can either inject in vivo to mice or apply it to cells and look specifically on newly synthesized protein. So looking on the mice neuromuscular junction, you can see in red, it's a transgenic mice of chat rosa. That means basically that only the motor neurons are stained in red. And you can see in green, the OPP, the newly synthesized proteins at the NMJ, and injecting it into the muscle, we saw that there are newly synthesized proteins at the NMJ, again, far away from the cell body. And as long as I know, this is the first evidence showing significant local protein synthesis at the NMJ. People have shown a lot of local protein synthesis along the sciatic nerve, along axons after injury, but not at the NMJ. So we thought that there is local protein synthesis at the NMJ, but what happened when we induce the TDP mislocalization to the axons? So looking both in vitro at our culture, we thought that there is significant decrease in the locally protein synthesis, both along axons and in our in vitro co-culture system, as well as in vivo. When we injected it to the mice and looked on sciatic nerve, we can see significant decrease in local protein synthesis along the axons and in the neuromuscular junction. So TDP condensate lead to decrease in local protein synthesis along the axons and the NMJ. We next wanted to understand, so if what are the protons or what protein changing their translation upon this TDP mislocalization. And for that, we did a proteomic screen of sciatic nerve with the TDP condensate compared to the control. And what we found is that many nuclear encoded mitochondria genes or proteins are missing, are downregulated. So maybe a quick reminder, mitochondria or have something like around 1,000 proteins. Most of them are encoded in the nucleus. Only about 30 are from DNA of the mitochondria. And what we see here, that the TDP condensate downregulate many of the mitochondrial proteins along the axons. When we looked on the RNA level, we didn't see significant change, and actually we thought that the levels of the RNA are a bit above, suggesting that this RNA is accumulating but cannot be translated. So the proteins level going down while the RNA is not changing, or maybe a bit go up. So what does it mean? What is the functional meaning of downregulation of nuclear encoded mitochondrial gene in the axons? And whether, so to answer that, we first inhibit protein synthesis in the axons, either using amizomycin or sodium arsenite, which causing stress and stress granules. And we saw that the inhibition of translation in these two ways can affect the mitochondria function using TMRE, which is a dye that can dye the membrane potential and is a marker for functional mitochondria. We also saw that, that in the axons that express the TDP, when we treat them with anisomycin that inhibit the protein synthesis and disturb the function of the mitochondria, the recovery of those mitochondria along the axon is decreased, suggesting that, again, that TDP condensate inhibit local protein synthesis and interfere with the recovery of this protein upon stress. But does NMJ need mitochondria? Does the axon actually need mitochondria? And the answer is yes. And to show it, we use a mitokiller red probe, which is that by light, we can activate specifically toxicity of mitochondria. So it's a gene that we transfect only the neurons, and when we activate it, it's release ROS, it's release stress that damage the mitochondria. And when we damage specifically the mitochondria at the pre-synapse of the neurons, we saw decrease in the muscle connectivity, in the NMJ activity. We also saw that when we specifically inhibiting local protein synthesis at the pre-synapse, and here we did it again with our microfluidic chambers using poromycin-resistant muscles and adding poromycin to the NMJ, we saw that there is decrease in the NMJ activity, suggesting that the NMJ activity depends both on local protein synthesis as well as on mitochondria. And finally, when we inhibit local proteins of axons, we saw that the axons that represent this TDP condensate have decreased the ability to regenerate and degenerate fast. So I show you that TDP condensate can regulate local protein synthesis. It can specifically regulate nuclear encoded mitochondrial proteins, and this is essential for the maintenance of the motor units. But what will happen if we recover the TDP mislocalization? And using this DOX system, we can do that. So when we restore or stop or starting adding again the DOX to the culture or to the mice, we can see that the TDP is getting out from the axons again, here in western blot of sciatic nerve or by sciatic nerve axons. So we can recover and restore TDP mislocalization along the axons, as well as its TDP condensate at the NMJ. And when we restore this, we can also restore the ability of the NMJ to locally synthesize proteins, both in vitro and in vivo. Again, suggesting that TDP condensate at the NMJ is essential for protein synthesis. And what does it mean? We wanted also to look specifically on nuclear encoded mitochondrial gene. And for that, again, we injected the OPP to the NMJs. And you can see here in this movie, the neuromuscular junction. Again, in red is the motor neurons, the presynapse. In green is the example of COX-4 of a nuclear mitochondrial gene that is found at the NMJ. And if you look on the co-localization of the OPP, you can see that upon TDP condensate at the NMJ, there is a down-regulation of newly synthesized COX-4 that is repaired. And with that, I will finish. I will just say that also functionally, when we restore the TDP, we can see back the activity of the NMJ and the morphology in vivo. So the model that we're suggesting is that TDP43 mislocalization to axon generate a gain of function that contributes to NMJ disruption by inhibiting local protein synthesis. And I will just finish by giving thanks to Topaz and Ariel, our really great PhD students that together with the lab push this project, and to you, other many collaborators and funding agencies. So thank you very much, and I will be happy to I guess there's no time for questions, but Mail will be happy to answer. So thank you.
Thank you very much, Eran. And without further ado, we'll move to our next speaker, who is Daniel Harries from the Hebrew University. And the title is How Crowding and Cellular Solutes Determine Aggregation and Phase Separation. Daniel, please.
Sure. Thank you. I'm really enjoying this meeting. I'm learning a lot. So mine will be a physical chemist's perspective on this topic. And what I'd like to tell you about today is about the effect of crowding on these condensates, and more generally on phase separation in macromolecular systems. So when we think about crowding, we can think about many different length scales, from very large objects to the very small. And on all these length scales, we can see that, that crowding profoundly influences self-assembly. So here is an example of needles that are vibrated, shaken, and when they are alone in a tray, they show this kind of random behavior. But once you add ball bearings to the mix, you see that aggregates start to form. This is the onset of a phase transition occurring simply because of this added crowding. we know that cells are highly crowded by macromolecules, and this can further impact these types of interactions that lead to these phase separations. So typically when we think about control of macromolecular interactions, we think about different kinds of physical parameters such as temperature, which in fact translates to the strength of an interaction, pressure, pH. But to these, we should add the complexity of the cellular environment that adds many other solutes to the system. And these can include inorganic salts, sugars, other metabolites, and also many macromolecules, protein, DNA, RNA, and so on. And then the question is how these different solutes affect, modulate, impact the interaction between the different macromolecules. And so in the context of liquid-liquid phase separation, we often think about plots like these that describe the coexistence as a function of composition. Typically, in these plots, we would show only one component, maybe two, if we go to more complex phase diagrams. But we know that, in fact, there are many other solutes in cellular milieu that we should consider when we think about these. So how do they impact this phase diagram? It could be something like this. Solution conditions can expand or diminish the size of the coexistence region within these phase diagrams. And one simple way in which this can happen is through solutes that are typically excluded, so they do not favorably interact with different components in the cellular milieu. A striking example are these tardigrades. They can do a fantastic feat once they are under stress, dehydration or other types of stresses. They can go into this dehydrated ton state. They accumulate very large amounts of a sugar called trehalose. Other tardigrades produce large amounts of intrinsically disordered proteins. And in all these cases, they are able to survive for prolonged periods of time with very little water, maybe 5% of hydration. And just by this accumulation of this added solute, they can maintain the function of the different cellular components. How is this done? Has been understood for a while. Here is an example of what different solutes, excluded solutes, ones that do not stick to the macromolecules, how they can change the size of an intrinsically disordered protein. And in all these cases, we see in terms of the free energy that there is a stabilization of the more compact state of this protein, and this effect becomes larger the larger the solute. And here is an example for many different solutes of varying sizes. And this is understood in terms of a depletion force. So the depletion force tells us that, at least according to the simplest model of Asakura and Osawa, tells us that there is a favorable free energy for the association of molecules, large molecules in solution in the presence of smaller ones, that is due to their exclusion. And when these two molecules come together, say the yellow ones, there is an added volume that is now freed. And so there is an added entropy for the solutes that are in solution. So this is a driving force. It's a solvent-mediated attractive force that is driven by a pure exclusion of small solutes from macromolecular surfaces. And this is indeed well understood, but then when we started to think about these problems, we realized that in fact the excluded solutes stand on one side of a very large gamut of different solutes, some of which can be attracted to macromolecules, some of which can be repelled from them. And on both sides of this very large spectrum of interactions, there is a possibility for stabilizing macromolecular complexes. On the one hand, by local variations in solutes around macromolecules, one can bring these macromolecules together. Basically, this is a type of bridging interaction within or between large macromolecules. On the other hand, by exclusion, you can also compact macromolecules through what I just described as the depletion interaction. So by varying the interaction between solutes and the macromolecules, one can stabilize, destabilize, or stabilize again, depending on how strongly these solutes interact with the interfaces of macromolecules. And this interaction can be modulated by physical parameters such as pH, salinity, and temperature. So all of this can be described in terms of theory. As a theorist, I'm allowed to show one slide of equations, but the theory for solvents and in general multi-component solutions has been around for a while, but by adding this possibility of local variations in solute concentrations around macromolecules, we can add this degree of complexity and put on one footing this whole spectrum of interactions. And so here is an example of a calculation that can explain the re-entrance from a collapse to an expanded and back to a collapsed state of macromolecules. This can explain experimental observations such as these, where intrinsically disordered proteins or other large polymers can go through this compaction and expansion transitions by locally accumulating added solutes around their macromolecules. Here is another example showing aggregation and phase separation of an intrinsically disordered protein in the presence of these types of excluded or included solutes, again, depending on the concentration and other solution conditions. So this can bring about a large type of, or a very complex type of phase diagram and very simple interactions, attraction and repulsions can bring about these kinds of complex phase diagrams with all its richness. But then there is another complication. So we were thinking about this coexistence region so far, but what if we go a little bit above this coexistence region into this supercritical region? And there, interesting things can start to happen. Specifically, you need not think anymore about macrophase separation. There can be subtleties. So things like oligomerization can be perhaps modulated by solution conditions. And this is what I'd like to tell you about now, because we know that some of the cellular components act as what are called hydrotropes. And hydrotropes are solutes that increase the solubility of hydrophobic or molecules that are hardly miscible in water. But in contrast to self-assembling systems, they do not themselves form mesostructures, so they are not exactly like surfactants, but they can increase the solubility of other molecules that are hard to solubilize. So recently, work from some of the people that are also attending this meeting has shown that, that in fact, ATP acts as a hydrotrope. And probably there are other molecules in cells that are acting in this way. And why are they interesting? So if it was just a molecule that is increasing solubility, perhaps they would go under a simple class of such molecules. But what we are finding is that these molecules have specificity. Despite their very weak interactions with macromolecules, they also hold specificity. So what do we mean by specificity in this case? If the molecules were non-specific, were acting non-specifically, it would be like these green molecules that are just solubilizing the molecules that are hard to solubilize, but they would increase the solubility of all molecules, all species in solution equally. All oligomers would be solubilized to a similar extent. However, a specific molecule would pick one oligomeric state. For instance, it might choose the monomer, as is shown here, or a dimer, and so on. Is that possible? Could a molecule that is interacting through weak interactions, could that still hold specificity? When we wanted to test this, we were thinking about a simple molecule that we could use. And our model that we chose was caffeine. Caffeine makes linear aggregates in solution, and as shown here. And we were testing this oligomerization process in the presence of sugar, different sugars. Specifically, we were also looking at trehalose that I mentioned before in the case of the tardigrades. So here are some interesting facts about what happens to caffeine in the presence of sugar. So the first thing that happens is that sugar decreases caffeine solubility in water. So it seems to favor the oligomeric state or the crystal state, and maybe is acting opposite to a hydrotrope. But here is the surprise, that sugars, at the same time that they increase aggregation, they impede oligomerization. And that sounds at first like a contradiction, because if something inhibits oligomerization, you would think it would also increase solubilization of the crystal state. But this is not what happens. How is that possible, that such a simple interacting molecule is responsible for these two opposite types of behaviors? And so by using molecular dynamic simulations and some other types of modeling, we could resolve the interaction of the sugar with caffeine. And it turns out that the sugar interacts favorably with the tips of the oligomer, but not with the sides. It is excluded from the sides, but is attracted to the tips. So it is solubilizing molecules that have more of the tip type of exposed surfaces, and is destabilizing anything with exposed sides of these columns. And so all in all, we have this opposing forces that lead to specificity. And we find that this hydrotope, sugar in this case, is acting in contradicting ways and then picks out one oligomeric state from all others. And so what I try to tell you about today are two types of ways in which the environment, the solution conditions can modulate macromolecular interactions. One is through the depletion force or the bridging interactions that can shift the coexistence up or down depending on the type of interactions. And the other is what happens in this supercritical region where oligomerization and fluctuations in sizes of aggregates becomes important. I'll end by thanking the great people who did all the work, and thank you for listening.
Thank you very much for the very interesting talk. Since we have 2 minutes more, if anyone wants to ask a question, please send it by the chat. But then I want to, and if I'll get some questions, I'll pay attention to that. I just wanted to know whether using your modeling for the first part of the talk, can you take components that we are interested in and try to foresee whether they could be in a condensate or they will be pushed out, like you said?
Yes, if there is some information on the type of interactions between these components, then it is definitely possible. So these models are fed by empirical parameters that are experimentally accessible. I mean, sometimes an experiment needs to be performed to find them, but they are derived from experiments. And if you know those interactions, then yes, definitely you will know what will be their composition within the condensates.
Okay, thank you very much. And if there are no more questions to Daniel, we'll move to the last talk in this session by Robert Tycko from NIH, Molecular Structure of Low Complexity Protein Assemblies, Insight from Solid State NMR, cryo-EM, and Simulations. Please, Robert.
I'm sorry. Okay, I had myself muted. Can you see my slides? Is that okay?
Now we can hear you. Thank you.
Okay, you can see it? Okay, great. So thank you very much for the invitation to participate in this very interesting meeting. It's a pleasure to be with you. And I'm going to be talking about a topic that's a little bit different from the topics you've heard about in previous talks, but I hope there's still some relevance to things that other people are doing. So I'll begin by just pointing out the people who have done this work. So this is a recent photograph from one of our group meetings back when our lab was shut down in April. And I hope you can see my cursor.
Yes, we see.
Okay, good. So the work I'll be describing is primarily work done by a current postdoc in my lab, Myung-Woon Lee, who's shown here, with assistance from Kent Thurber, a staff scientist in my group. And Masato Kato is a collaborator who's actually spent about 1.5 years on sabbatical in my lab, coming from UT Southwestern, from Steve McKnight's lab. And of course, we've been collaborating with Steve McKnight from UT Southwestern. He sort of introduced me to this problem of low complexity sequences. from RNA binding proteins and their biological importance. And we've been working together on that for the past 8 or 9 years with Stephen Masato. Okay, so we've been studying, what I'm going to be talking about are amyloid fibril or amyloid-like fibrils that are formed by low complexity sequences, in particular the low complexity domain of the FUS protein. And we've been studying amyloid structures for more than 20 years now, beginning with the amyloid beta fibrils that are associated with Alzheimer's disease, the amylin or IAPP fibrils that are associated with type 2 diabetes, and mammalian and yeast prion proteins. And these proteins have very diverse sequences, but they all form these long filamentous structures that look at least similar to one another in electron microscope images. So forming amyloid fibrils seems to be an almost generic property of polypeptide chains. If you look at their sequences, they are indeed rather diverse in their length and their amino acid compositions. But in many cases, an important aspect of their amino acid composition is that they contain a significant fraction of hydrophobic residues and hydrophobic segments. And as you'll see, in many cases, it's the hydrophobic interactions that really are driving amyloid formation. I'll turn on my video too. Okay. In the case of the FUS low complexity sequence, though, there are very few hydrophobic residues. There's no leucines, no isoleucines. There's one methionine, no phenylalanines. So when I first saw the FUS low complexity sequence, my initial reaction was it really shouldn't form fibrils. It should just be a soluble sequence. That turns out not to be true. So part of my own work is like really trying to understand why do sequences like this form fibrils. And to get insight into that, you have to investigate their molecular structures. So we've been studying molecular structures of other systems, other fibril forming systems for over 20 years. All of the amyloid fibrils by definition contain beta sheet structures organized or oriented in what's called a cross beta orientation, which means it's a beta sheet where the beta strand segments that form the sheets run approximately perpendicular to the fibril growth axis, and they're connected by interstrand hydrogen bonds, that's these green bars that run parallel to the fibril growth direction. And the original evidence for this kind of a cross beta structure in an amyloid fibril came from X-ray fiber diffraction data, originally by Eanes and Glenner at the NIH. My own lab is in the intramural research program of the National Institutes of Health. And actually these people also were in the intramural research program of the NIH, but back in 1968. Here's some more recent X-ray fiber diffraction data for the A-beta, amyloid beta, 40 amino acid, 40 residue amyloid beta fibrils, A-beta 40 fibrils from 1998. That's actually when we started working on amyloid fibril structures in 1998. And at that time, the only thing that was really known about their molecular structures was that they contain these kind of cross beta structures from X-ray fiber diffraction data. We've learned a lot since then, originally by doing solid state NMR measurements, which I'm not going to go into any details about that, but that's a set of techniques that my lab specializes in. One of the things that we learned early on is that these cross beta structures can either be parallel beta sheets or anti-parallel beta sheets, but in fact, the parallel beta sheets predominate, except in unusual cases, especially short peptides that form amyloid fibrils can be anti-parallel, but long protein sequences, let's say more than 20 amino acids, are almost always parallel beta sheet structures. And so we showed that back in this paper in 2000. This was work that was done by Oleg Sutkin, who's a visitor in my lab for a year or two, who actually got this whole research effort started. So I always like to give him credit for that. Thanks, Oleg. So here the residues are colored, green being hydrophobic, and you can see that the parallel cross beta structure lines up all the hydrophobic residues with one another, whereas in general, an anti-parallel structure would not do that. So if hydrophobic interactions are driving the fibril formation, it kind of makes sense that you would favor this parallel structure. And that turns out to be true. You also, by having a parallel cross beta structure, you also line up some polar residues with polar side chains, in particular glutamine residues. Their side chain amide groups can form long rows of hydrogen bonded rows that were originally pointed out by Max Perutz. He called those polar zippers. So polar zipper interactions can be important, and those are also maximized by this in-register parallel cross-beta structure. The bad thing about a parallel cross-beta structure is if you have charges, very charged residues, then you'd have strong repulsions among charged side chains, and that would blow the fibrils apart. So the actual structures avoid that in various ways, either by putting all the charges on the outside of the structure or by pairing up oppositely charged side chains. Now, an important aspect of amyloid fibrils is that they're almost always polymorphic. So the same amino acid sequence can form fibrils that look somewhat different from one another in electron microscope images, and that's shown here for two different polymorphs of the 40 amino acid A beta fibrils, the A beta 40 fibrils that we've prepared in vitro, just growing them from synthetic or recombinant peptide in vitro. We've also looked at brain-derived fibrils. I'm not going to talk about that today. But just subtle variations in the growth conditions can give you fibrils that look quite different, and they actually are different at the level of molecular structures. They have different solid state NMR spectra. In this case, the difference is whether you let the fibrils grow quiescently, so you just dissolve your peptide in the solution and then just let the fibrils grow over a period of hours to days, or as the fiber, or you dissolve your peptide in the same solution, the same buffer, the same temperature, and you just agitate the solution as the fibrils are growing. You just slosh the solution around, and that will give you fibrils that look different from one another. So you can ask me about that later. Why is that? What's the mechanism for that? But the point is that very small variations in growth conditions can give you fibrils that look quite different from one another. And then these are two, the molecular structural models. These are early models that my group developed. It's work of Ani Petkova and then later Anant Paravastu for these two different polymorphs. This is based on some electron microscopy and also on solid state NMR restraints. And these are cross-sectional views, so the fibrils are coming out of the screen towards you. And again, green side chains are hydrophobic. So again, you can see you have these clusters of hydrophobic residues. Hydrophobic interactions are driving the fibril assembly in this case. So that's just some of the background. Stabilized by hydrophobic interactions, one thing that means is that their stability increases with increasing temperature. Right? So that means the solubility decreases. The fibrils become more stable at higher temperatures. That's actually the opposite of things like the FUS low complexity sequence, where they tend to dissolve at higher temperatures. Another aspect of the hydrophobic interactions is that it probably contributes to the polymorphism. There's many ways of just putting hydrophobic side chains next to one another that aren't very specific in terms of conformations and structures. And similar remarks apply to other systems, other fibrils that are involved in other neurodegenerative diseases, alpha-synuclein, tau fibrils. These are recent cryo-EM structures, again, cross-sectional views with the fibrils coming out of the screen towards you. Two alpha-synuclein polymorphs. In this case, the structures were solved by David Eisenberg and his collaborators at UCLA. Two of the tau fibril structures from the MRC lab, Shores, Shiris, and Michel Goder. These are derived from brain tissue. So again, you have this polymorphism. If you look at these structures in detail, you'll see that they're actually stabilized primarily by hydrophobic interactions. Now, the FUS low complexity sequence is different. Again, there's not much hydrophobic there. It's mostly serines and glycines with some glutamines and tyrosines and threonines thrown in. I would have expected that this would be a rather soluble kind of a polypeptide, but it's not. These sequences form fibrils that look very similar to the A beta or alpha-synuclein or tau fibrils. One surprising thing is, the interesting thing is that they have very uniform amino acid compositions. In the case of this FUS low complexity sequence, the amino acid composition is uniform over the entire length of the sequence until you get to the very C-terminal end where it becomes more glycine rich. But basically the first half and the second half of the sequence are almost identical. So I would have guessed that there would be many different ways. If this kind of a sequence is going to form fibrils, there would be many ways for it to form fibrils. It would be highly polymorphic because there's no, a priori, just looking at it, there's no reason to pick out any one particular sequence as being the most fibrillogenic or amyloidogenic or fibril core forming segment of this kind of a sequence. It could be, the core could be anywhere, it seems. But then when we actually did experiments using solid state NMR, we found that in fact, these fibrils are not polymorphic. In fact, under our conditions, we always get the same fibril core structure that's formed by residues 39 to 95. So that's this segment shown here. Turns out when you have this full-length FUS low complexity sequence, it's that underlying segment that forms the fibril core. We can tell that from solid state NMR. Again, I'm not going to go into the details. That was published back in 2017. It's the work of Dylan Murray, a former postdoc in my lab, who's now at the University of California at Davis, setting up his own lab. And again, collaborating with Masato Kato and Steve McKnight and other people in Steve's lab. This is what the structure looks like in cross-section. I'll just point out, I won't go into all the details, but I'll point out it's basically an S-shaped structure, or maybe a backwards S, the way I drew it here. It's an S-shaped core structure. So why is it that that particular segment always, we've made many samples independently, we always get the same solid state NMR spectra and the same core structure? Is it that other segments for some reason are incapable of forming good fibrils? So I'm going to talk about that, but before I do, let me just point out a couple of things. As I said, amyloid fibrils are usually polymorphic. The one case that I know of really where the fibrils are not polymorphic is the het S fungal prion fibrils. This is the structure solved by the ETH group, Beat Meyer and Roland Rieck and their colleagues some years ago. And we've also looked at the het S fibrils. We get the same thing that they do. It doesn't matter. You always get the same structure. It's clearly non-polymorphic. And this is one of the amyloid-like fibril structures that really has a well-defined biological function in the phenomenon of heterokaryon incompatibility in Pedospora fungi. So this makes me wonder if maybe the absence of polymorphism in the FUS low complexity fibrils may be some indication of biological functionality. That's just a question, but it is an interesting observation. And then I just want to show this kind of movie, if I can do this, just to give you an idea. So we have this fibril core structure that's formed by residues 39 to 95, but then there are tails that we know from the NMR are disordered. So it's a core structure surrounded by these long disordered chains, and that's going to be important at the very end. Okay, so why are other segments incapable of forming good fibrils? Well, no. So based on some work that Masato Kato had done, we began looking at fibrils formed by either the first half of the sequence, residues 2 to 108, the N-terminal half, or the second half, the C-terminal half, residues 110 to 214. Both of those halves by themselves form nice fibrils. Based on solid-state NMR spectra shown here, when you just use the N-terminal half, you get the same solid-state NMR spectrum as you do for the full-length sequence. So you get the same core structure. So that makes sense. So the core structure is in the N-terminal half, and that's what you get when you just have the N-terminal half as your construct. But the C-terminal half can also form very nice fibrils. It has a different solid-state NMR spectrum. It's a different sequence. If you look at those sequences, again, the amino acid compositions shown here are rather similar. Again, both cases, it's mostly glycine and serine. It has some about 20% glutamines, about 10 or 15% tyrosines. They're quite similar. These fibrils look extremely homogeneous to me. These are negative stain TAM images. These are very homogeneous looking fibrils with a very regular twist. So I thought these might be suitable for cryo-EM measurements to investigate their structure. And so we pursued that. And we've, cryo-EM structures of amyloid fibrils, this is something that's was impossible 5 years ago, but now it's possible. And over the past 3 or 4 years, there are many examples of that. I already showed the examples for alpha-synuclein and tau. We've learned how to do this. We've gotten to be pretty good at it, I would say. And so Myung-Woon Lee, who I mentioned at the beginning, currently a postdoc in my lab, worked on the cryo-EM, assisted by Kent Thurber, staff scientist, and former postdoc Eugenie Ghosh. And this is what we came up with. So these are cryo-EM images. These are one of the 2D class averages. It looks very nice. This is the density, cryo-EM density map, 2.6 angstrom resolution for the density map, which is quite good. It's a structure with now twofold symmetry, actually screw symmetry about the fibril growth axis. And this work is currently in press. It should come out very soon. The resolution of the density is sufficiently high that you can unambiguously fit the chain into the density and construct molecular models. So this is what the model looks like. Has some very interesting features, a nice so-called steric zipper interdigitating side chains at the interface between the two halves, the two cross beta subunits. I don't have time to go through all of this. It's formed by, the core is formed from the density. We can tell that the core is formed by the sequence shown here, residues 112 to 150. And then the C-terminal part of it is still a disordered tail. This core forming segment now forms a U-shaped structure. We saw an S-shaped structure. This is a U-shaped structure. There's a disordered tail at the C-terminal end. You can see that in the cryo-EM density just as a halo around the core. You can also see in solid-state NMR spectra that there's a lot of signals from glycines, broad disordered signals from glycines. So part of that disordered tail is actually statically disordered. From other kinds of solid-state NMR measurements, we can tell that part of it is also highly dynamic. So it's a mixture of static and dynamic disorder for the C-terminal tail. We want to get information about the interactions that stabilize this core. So we did some long molecular dynamics simulations in water. You can see where the water is going. You can see how this hydroxyl bearing and polar side chains interact with one another. And from analyzing this in detail, you can see that there's a very complicated network of hydrogen bonds that stabilizes this core, including hydrogen bonds between side chains, hydrogen bonds from side chains to backbone groups, hydrogen bonds between the two different cross beta units that hold the two together, these polar zipper hydrogen bonds along the growth axis. Is a complicated network of hydrogen bonds. You can also see that side chains are moving around. Tyrosine residue side chains are flipping. Glutamine side chains, some of them are flipping. Some of them are more permanently held in place by these polar zipper interactions. So I'm running out of time, so I'm rushing through this, but this is all will appear in the paper that's coming out soon if you're interested. And the main point, it's qualitatively different interactions. These hydrogen bonding interactions are very different from the hydrophobic interactions that stabilize something like the A beta structure. So now we know that at least, this is my last slide, now we know that at least two segments of this fused low complexity sequence can form a cross beta fibril core. But in the context of the full length sequence, the N-terminal core is favored always. As I said, it seems not to be polymorphic. So why is that? If there's multiple possible cores, why do we always get the N-terminal core? So is it because the N-terminal core, for some reason, is just a better structure? It's more stable inherently. Well, I think that's not really true. So under the conditions of our experiments, you can measure the equilibrium solubilities of the fibrils formed by either half, the C-terminal half or the N-terminal half. Masato Kato did this when he was on sabbatical in my lab. And they have very similar solubilities, which means that the inherent stabilities of the two cores are essentially very, very similar. It's a very small difference. So is there some other explanation for what determines what the core forming structure is? And now I'm just going to speculate about something. So I'm closing with a speculation here. So it's important, I think, to realize that the two cores involve just a small fraction of the total amino acid sequence, less than 30% of the total sequence. And there are very long disordered tails representing 70% or more of the total sequence. So what the tails are doing could be important, actually. Now, when you take a long disordered tail, like a 100 residue chain, it likes to form a big random coil. But then if you attach those tails to a cross beta structure, then you're forcing one of their ends to be about 5 angstroms apart. You're bringing them very close together, and then they start colliding with one another. And that reduces the conformational flexibility, the conformational freedom of the disordered tails. So there's an entropic cost associated with the tails, with bringing these disordered tails close together in a fibril structure. Okay, and that entropic cost from simple estimates can be quite large. It can be 5, 10 kcals per mole. And that can be a larger effect than are competitive with the actual stabilizing interactions within the core. Now if you take the full length sequence and you take the N-terminal core, this is what we actually observe. That because it's an S-shaped structure, it puts the N-terminal and C-terminal tails apart from one another. In the alternative structure, which doesn't actually exist, where you have a C-terminal segment forming the core. You have both C-terminal and terminal tails that, because of this U-shaped conformation, are forced to be close to one another. So you get even additional restrictions on the conformational flexibility, the conformational freedom of the disordered tails in this alternative structure, which doesn't actually exist. So maybe it's actually the properties of the disordered tails that are selecting the N-terminal core as being the experimentally observed core. So this is just speculation. I'm going to stop there. Thanks very much for your attention.
Thank you. And I guess we should take a break now. Yes, boss? How long, Eran? It's you?
Yeah,
For how long? 5 minutes or 10 minutes?
Let's go for 5 minutes. So we are back on schedule at 6:30.
Okay.
Good.
Thank you, all speakers of the session.
Great. Rick, I think you can start whenever you're ready. Thank you for taking over.
Happy to do so, Eran. Hello, everyone. I'm Rick Young. We're going to begin with Tanja Mittag. She's going to talk about valence and patterning of stickers in intrinsically disordered proteins determines their phase behavior. Tanja?
Yes, thank you, Rick. I'm very happy to be able to share my work with you, or our work with you today. Just need to find here how to share. Do you see my slides now?
Yes.
Okay, perfect. All right. So, as Rick said, I want to talk to you about how phase separation is encoded in intrinsically disordered protein regions. And we've seen this a lot yesterday and also today, that often disordered regions are sufficient for driving phase separation, as here, for example, in HNRNPA1, which is one of the systems that we work on. But really early on, it became very obvious that disordered regions play important roles in phase separation. And I want to briefly remind people that intrinsically disordered proteins or protein regions fail to adopt a single folded structure. And instead, there are many different conformations that interconvert. And I want to ask the questions today: how is the driving force for phase separation encoded in IDRs? And can we understand their phase separation quantitatively rather than saying, does it phase separate or does it not phase separate? Because of course, there is a certain driving force and that determines the saturation concentration of the system. And I want to use the opportunity to put away with the myth that all IDRs mediate phase separation. So what types of interactions drive liquid-liquid phase separation? We understand this very well in typical multivalent domain motif systems, first through seminal work by Mike Rosen's lab. And in these systems, multiple modular binding domains are linked in one protein, and the binding partner has multiple short linear motifs that interacts with these domains. And that leads to the formation of these large non-covalently networked complexes that have a lower solubility and therefore lead to the formation of these networked droplets. But and it's clear that the higher the valence of these multivalent proteins, the lower is the saturation concentration, as we can see here in these in this schematic phase diagram. But how does this work in IDRs? And I think the problem is that we don't really understand what the interactions are that drive phase separation and how strong they are in certain parts of the sequence. Whereas in these folded systems, even if the interactions may be very weak, we have a physical picture from structures and we can determine microscopic interactions of these individual interactions. And that allows us to really understand how the phase diagrams are encoded. And I would say if we knew this in IDRs, then we could also understand the phase diagrams of IDRs better. And we would probably expect that the valence there also determines the phase diagrams of these IDRs. And Rohit Pappu has put a framework forward, the so-called stickers and spacers framework, where we think about these IDRs in exactly this kind of way, where we have adhesive elements that we call stickers and spacer elements that connect these stickers. And this helps us understand conceptually how IDRs phase separate. So we're building on work from Julie Foreman-Kay and Andy Baldwin, Michael Rosen, Pappu, Alberti, and Hyman, and they have taken mutational analysis or chemical modification steps of certain residue types to understand their contribution to phase separation. We wanted to take a little bit of a different approach and do an unbiased search for stickers in the sequence. And the idea that this relies on is that the same interactions that drive phase separation via intermolecular sticker-sticker interactions should also be satisfied intramolecularly and should lead to the compaction of proteins in the dilute solution. And this project was in collaboration with Rohit Pappu. Alex Holhaus, who was a postdoc at the time, now has his own lab at WashU, was driving the daily work on his side. On my side were two postdocs, Eric Martin and Ivan Perrin. And Anna Bremer and Mina Frag helped out, and they are really driving the follow-up projects now. we directed this work towards the low complexity domain of HNRNPA1, which is sufficient for driving phase separation. And we use this, this protein because it shares a lot of sequence features with other low complexity domains of RNA binding proteins, for example, in the FET family. And if you look at the sequence, we have seen this now a couple of times and of similar proteins, and Rob Tycko just talked about FUS. So here we have a similar sequence where we have glycine and serine making up 55% of the sequence and 6 amino acid residue types making up 86% of the sequence. And so I describe this sequence as mostly being small polar residues and then interspersed aromatic residues and some charged residues. So we suspected that this sequence was compact. We tackled this, Eric Martin tackled this by size exclusion coupled small angle X-ray scattering, which gives us information on the size of protein, size and shape of protein. You can see this best in this so-called normalized Kratky plot, where the plateau here directly tells us something about the size of the protein, especially about the scaling exponent, which tells us how compact or expanded a protein is and how well it likes to be solvated. And here we have the data for the low complexity domain of HNRNPA1. And the scaling exponent of 0.45 was really very compact for IDRs and showed that this protein doesn't really like to be solvated very much and probably prefers to make interactions with itself. The protein is intrinsically disordered, of course, and we can still get atomic information from NMRs or resolution specific information from NMR spectroscopy. And we took advantage of this by doing relaxation experiments where we have here these R2 relaxation rates that tell us something about the dynamics of the residues in the chain. And what we saw was that there were these excursions, these higher R2 relaxation rates that were really a deviation from what we expected, where we expect a plateau across the chain and just some tapering at the end. And if we said, okay, we have these excursions and we make them explicit, and then we saw that a lot of these clusters were very close to aromatic residues in the sequence. And this model is consistent with clustering of aromatic side chains. And to follow up on that, we looked for this clustering directly for contacts of aromatic side chains with each other. In NMR spectroscopy, you look for NOEs, which come from direct transfer of magnetization through space. And we could indeed see NOEs between phenylalanine and tyrosine residues, or here tyrosine residues and phenylalanine residues. So they are indeed clustering in space. Alex Holhouse and Rohit's lab did all-atom Monte Carlo simulations with the absinthe implicit solvent model. And here we're highlighting the aromatic residues, and perhaps you're seeing already that they are interacting quite a bit. The simulations were in very good agreement with our SAX data, which gave us some certainty that we could actually interpret them in a molecular manner. If we here analyze the contact order, which is a measure of how often certain residues in the sequence or all residues in the sequence interact with other residues in the sequence, you see these spikes, and that nearly all of these spikes are located at the aromatic positions. And here in this network analysis, we see with how many other aromatic residues each aromatic residue interacts. And we see that there are not only pairwise interactions, but basically that each aromatic residue can once in a while interact with all other aromatic residues. And so this really pointed to aromatic residues as the stickers in the sequence. But we wanted to know whether we only saw them interacting because the protein was compact and the aromatic residues are bulky and so they see each other, or whether they were actually driving the compaction of the protein. And so we made a number of variants in which we changed the number of aromatic residues, either added 1/3 aromatic residues, removed 1/3 or 2/3 of aromatic residues. And we measured the size of these protein variants again via small angle X-ray scattering. And indeed, we see that increasing the number of aromatic residues compacted the protein further, decreasing the number of aromatic residues decreased the compaction of the protein, showing that these stickers are aromatic in nature, and they give rise to cohesive interactions that determine chain dimensions. Now, that was good. So we knew what seemed to be the most important stickers. We wanted to know whether they're really, these are the main drivers of phase separation in this protein. But also it gave us an opportunity to know what the microscopic affinities of these aromatic residue interactions were. And this is based on these experimental RG values of our variants. And Alex and Rohit parameterized a coarse-grained model that uses a single bead per residue. We just have two residue types, stickers and spacers. And they parameterized the strength of the sticker-sticker, the sticker-spacer, and the spacer-spacer interaction strength. And as you would expect from a good model, we see that, with this simple model and a single chain setting, we get very good agreement of the experimental RG and the RG value from this model. So that's good, which meant we could now put a lot of chains into boxes and observe phase behavior. As you can see here for wild type, ERA minus and ERA minus minus. And you see a difference in their phase behavior very clearly, right? Importantly, it's also possible to determine concentrations inside the droplets and outside of droplets. And if you do this as a function of temperature, you can map phase diagrams. And we look here, for example, at a given temperature or at all temperatures, the ERA plus saturation concentration is lower than the wild type saturation concentration, which means ERA plus has, of course, a stronger driving force for phase separation. Also means overall, if we interpret this, the higher the valence of the stickers, the larger is the driving force for phase separation. Absolutely equivalent to the domain motif type system, where the valence determines the saturation concentration. Now, a bit of a problem with these phase diagrams is that we have a simulation temperature here instead of an absolute temperature, a volume fraction instead of a molar concentration. So we need to bring this back into the laboratory frame to make use of these phase diagrams. And we did this via experiments, of course. And if we want to measure phase diagrams, we better know that we have equilibrium phase separation and it's reversible. We're looking here at decreasing the protein concept, decreasing the temperature, and you see the appearance of these droplets under the microscope. This also results in scattering. If we now increase the temperature again, the droplets go away. So all of that works well. These droplets are also very nicely liquid and fuse well, which means we can make large enough dense phases to measure their concentration and the coexisting light phase concentrations. We also wanted to know what the coexistence behavior is close to the critical point to be able to better define this phase behavior. And so Ivan did some cloud point measurements at pretty high protein concentrations, but that were nicely reliable. Now, these experimental data now allow us to bring the stickers and spacer simulations into this laboratory frame. And we now had a fully parameterized stickers and spacers model to describe this phase behavior of the wild type protein. This also fit very well to the phase behavior of our other variants. And it also explained why we never saw ERA minus phase separate, because the critical point is actually below freezing. So this pretty simple stickers and spacers model reproduces the experimental phase diagram of all variants, which is nice. And we wanted to know whether we could also transfer it to other proteins of similar type. So we turned to the literature where there are experimental values for the FUS low complexity domains with variants in which 2 or 4 aromatic residues were removed. And there was cloud point data. And Alex's stickers and spacer simulations gave us a theoretical or computational stickers and spacers cloud point data, and there was a very good correlation. And this result points to the transferability of this pretty simple stickers and spacers model to other low complexity domains. So the valence of stickers and their pairwise interaction strength determines the phase behavior of IDRs. This is the result that we're seeing, right? I also want to point out that it gives us a physical picture of what's going on inside these droplets. There is a condensate spanning network of protein molecules that are enabled by physical cross-linking of stickers. And I nicked this image from Rohit because I think it gives us an explanation for the size threshold of the nuclear pore complex for passive diffusion through the nuclear pore complex that was mentioned yesterday. So this meshwork has a certain size and it allows diffusion of molecules through it, and larger molecules need transport receptors to compete with these aromatic, in case of nuclear pore complex, of phenylalanine interactions to let it go through. We also, in this model, explicitly consider the positions of the stickers in the coarse-grained model, not just their valence. And of course, we had noticed that the position of the stickers is actually pretty uniform in the sequence. We went on to quantify this with this value omega aromatic. That is 1 if the aromatic residues are segregated from all other residue types in the sequence, and goes towards 0 if they are perfectly distributed in the sequence. And it turns out that the wild type is at the 99.9th percentile of uniformly distributed aromatic residues compared to sequences with a random distribution of sequences with the same composition. And so we wondered why that was important and why there was this evolutionary pressure for this uniform distribution. And we made a variant in which we segregated aromatic stickers in patches, and of course also a control where they were uniformly distributed. And it turns out this patchy variant makes amorphous aggregates, while the wild type and also the perfect variant make nice and liquid droplets. And so it really showed that the increased clustering of stickers led to some sort of super sticker that then led to very long interactions, very interactions with a long lifetime that changes the material properties of these assemblies. And so the spacers are not just connectors, they're actually important for diluting the interaction strength of the stickers and for driving solvation. This kind of linear distribution of aromatic residues in disordered proteins is pretty typical, actually. And we saw this a lot, for example, in other RNA binding proteins, but also in proteins related to vesicular trafficking and signal transduction. But there are also counterexamples, for example, ex vivo, which is an important component of Balbiani bodies, has a high segregation of aromatic residues. And it, of course, turns out that the Balbiani bodies make solid condensates. So nature may also be using these properties to tune assembly properties. So in conclusion, we can identify stickers in low complexity domains in an unbiased manner from experiments on dilute samples, which is useful if you don't have to actually look into the dense phase. Aromatic residues act as stickers in these Prion-Like low complexity domains, and they mediate distributive cohesive interactions. And linearly distribution of these aromatic residues promotes liquid-liquid phase separation over amorphous aggregation. And the spacers are important to dilute the interaction strength of stickers and promote solvation. Other sequences may have other sticker types, and the spacers are important for influencing the phase behavior. And these are things that we are working on right now to make models that can really explain phase separation of all types of IDRs. And I want to come back to the point that not all IDRs have a strong driving force for phase separation. We can, of course, mutate away stickers, but of course, there are lots of natural IDRs that actually are very solubilizing. And before phase separation became a thing to understand biology, a lot of people thought that IDRs were always very soluble and expanded. I want to take half a minute to make one more point, or to at least have have some share some thoughts on what the structural features are of sticker-sticker interactions. Are these condensates formed by the cross-beta interactions with structured backbones? We saw beautiful structures from Rob Tycko's lab in the last talk. Or do these chains remain disordered? And perhaps these interactions are driven via side chains. And there's data on both sides, right? The NMR spectra of dense phases that we saw from Julie Foreman-Kay's lab yesterday, and here I show you a spectrum from my lab, point to a lack of structuring. When we look in the dilute phase, really only the sticker residues are broadened and not whole motifs, pointing to their specific importance of these interactions. But perhaps cross-beta interactions are very transient and that thus we can't observe them by NMR. That's, I think, possible if there are many of these motifs that are competing with each other, as suggested, for example, by David Eisenberg's analysis of LARCs in IDRs. And but then we need to think about that LCDs can form liquid droplets and they can form hydrogels and fibrils. And if the same interactions mediate the formation of both, how does this switch between these two states occurs? And so I think it's useful to consider whether these kinds of sequences can encode two different types of assemblies and they are being formed by two different types of interactions. And with that, I'd like to acknowledge people in the lab who did the work. I pointed them out as I went along. I also mentioned, of course, that this was a collaboration with Rohit Pappu's lab, and I acknowledge funding, and thank you all for your attention.
Tanja, thank you so much for that. We're now going to move on to Itamar Harel, who's going to talk about the identification of protein aggregates with prion-like and phase separation properties in the aging vertebrate brain.
Thank you very much. I'm just going to share my screen. Can you see it okay?
Yes.
Great. So thank you very much, Rick. Okay, so aging is the primary risk factor for disease. This includes heart disease, Alzheimer's, cancer, stroke. Something happens in the 4th or 5th decade of our lives that the incidence of these pathologies increase. And the notion is in the aging field is that maybe we don't need to treat each and every pathology independently. Maybe we can actually slow down the rate of aging and delay the onset of all these pathologies at the same time. And this could truly revolutionize healthcare. So how do we study aging in the lab? In the past 20 to 30 years, these simple organisms, yeast, worms, and flies, dominated the experimental aging field, and for a good reason. They're short-lived, relatively, and they have superb genetics. But if you want to ask these questions or explore these pathways in a vertebrate context, there's a limitation because mouse and zebrafish live, give or take, the same lifespan of a grad student. So we have to bridge this gap, and this is the reason we and others have decided to use the naturally short-lived vertebrate, the turquoise killifish. So this fish lives in seasonal water ponds in Africa, in Zimbabwe and Mozambique. This is me in 2015 collecting some samples. And it is the shortest lived vertebrate that can be bred in captivity. There are multiple species with different lifespans. But more importantly, it displays all the human-like aging disease and phenotypes we see in ourselves, we see in mice, cats, dogs, and any other vertebrate, including decline in cognitive functions, cancer, decline in fertility, sarcopenia, kyphosis, and others. And conserved lifespan manipulations, such as dietary restriction, were also able to extend the lifespan of killifish. But what was gravely missing, besides the fact that it's a very attractive looking fish from Zimbabwe, is good genetic tools and genomic tools. So this is what I did during my postdoc. We developed genome engineering in this fish, and there were also 2 killifish genome projects that followed, as you can see cited below. And overall, the pipeline of generating new lines was quite efficient and rapid, thanks to the fact that these fish also experience one of the fastest sexual maturities across vertebrates, which happens after about 2 to 3 weeks. So this is an example of a transgenic line expressing a red fluorescent protein in the heart. We grow these fish in regular water recirculating systems, and there's about 30 to 40 labs around the world with steady killifish already, and this is a very vibrant and expanding community. And this is a course I'm teaching every year in the States. So I'm going to give you one example of what we were able to do with this fish. You can see that these are the hallmarks of aging, and we targeted many of these pathways, but I'm going to focus only on TERT, and we tried to replicate telomerase syndrome. And the reason was that telomeres in mice are actually quite long. As I mentioned, the lifespan of mice is quite long experimentally, and this brings us to the fact that exploring the link between aging and telomere syndrome is challenging in the lab. So we targeted TERT, telomerase reverse transcriptase, and we were able to recapitulate the phenotype that is seen after 4 or 5 decades in humans as early as about 3 to 4 months. And you can see, for example, a complete ablation of the gonad from germ cells, gut villi atrophy, and many other phenotypes. So overall, we have a model, a genetic model, that is quite versatile, high throughput, and we can use it to model or explore aspects of aging and disease, which are currently challenging to study in the lab. So now I'm going to shift gears and talk about protein aggregates and phase separation. And what we did here is, first of all, this was a fantastic collaboration, still is, which the majority of the work was done still when I was a postdoc under the supervision of Professor Anne Brunet at Stanford. And this is a great collaboration with Dan Yarosh, also at Stanford, and his PhD student, Yuan Chen. Also the lab of Sanchez Alvarado, and my own lab currently at the Hebrew University with 2 students working on this as well. So we've heard today and yesterday about protein aggregation, and some talks also talked about TDP-43 and ALS, Parkinson's and Alzheimer's, and we know that there is a tight association between protein aggregates and these neurotoxic pathologies. And some of these aggregates are actually proposed to propagate in a prion-like manner. What we don't know is whether there are, or what is the nature of protein aggregates that can potentially occur with healthy aging, and whether these aggregates can also maybe propagate in a prion-like mechanism. And this is the question we set up to explore in this project, and this is the experimental design. So we took 3 fish in each group, 3 young, 3 old, 3 telomerase deficient fish for an actual additional genetic insult. We isolated the following organs: brain, gut, testis, heart, skin, muscle, liver, and then we purified 2 samples, 2 fractions, one which we call the whole cell lysate, including the entire proteome of the tissue, and the other one, after a series of centrifugations and a sucrose cushion, we isolated the heavier aggregate-like fraction. Then we labeled them using TMT and used mass spec to identify the protein abundance. So now what we can do is basically calculate propensity, which means which proteins aggregate more in old individuals compared to younger ones if we normalize the abundance by the whole cell lysate. And what I was curious to see is there something that basically binds them together? Is each and every organ unique, has a unique feature, and what actually is common among all the proteins which can aggregate with age? And the first thing we tested was a prion, or enrichment for prion-like domain, and we used the plaque algorithm. And in blue, you see all the proteins which aggregated more with age, and this is about 25% of them are enriched for prion-like domains. And this is compared to what you see in red, which is the total proteins which aggregated in the brain, no matter if it was in young or old. So we see an age dependency in this enrichment. And if we calculate this abundance throughout the organs of the fish, we can see that this is something which is unique for the brain. This is the hits that we were able to detect, the top hits. And LLR represents, as you can see over here, LLR represents the prion-like domain. And the higher, the more positive the score is, the higher the chance that this protein, or the higher confidence this protein can truly behave in a prion-like manner. So you can see over here, I'm focusing on DDX5, which has a very, very high prion-like score. And we were curious to see whether this protein truly aggregates in the fish. So we generated 2 antibodies for the N terminus and for the C terminus. And we saw that the expression of DDX5 is via these small puncta in the nucleus in the young. However, these puncta stopped being nuclear localized and became more cytoplasmic with age. To test this in a different way, we generated a DDX5 fusion protein with GFP, transgenic fish, and we also saw these tiny puncta in the brain. And by staining sections in mouse brains, we also were able to show that this replicates also in mammals. So we identified this DNA, DDX5, which is an RNA helicase, actually responsible for the splicing of tau. And we showed you, we were able to show that basically it does seem to kind of aggregate and more in a cytoplasmic fashion with age, both in fish and in mammals. So how can we systematically evaluate this in a structure function kind of way? So this is how DDX5 looks like. It has 2 helicase domains, one which is ATPase or ATP binding. And at the C terminus, we see that it has a prion-like domain, an RGG domain. And also if we use disoprod3 as a predictor for disordered regions, we see that it has a disordered domain or intrinsically disordered region at the C terminus as well. So I'm going to focus now on 3 versions that we are testing. One of them is the full length. The other one is the delta prion-like domain or the delta PRD. And the third is the delta IDR, the deletion of the intrinsically disordered region. So if we use a regular phase separation assay, which Ewan performed by purifying the protein, we can play with protein concentration or salt concentration and get this phase separation diagram. And you can see the nice droplets formed. If we delete the prion-like domain, we see that there's a slight shift towards the right. So the prion-like domain is important for the phase separation. And as people have shown today and yesterday, you can see that the deletion of the delta IDR completely abolishes the phase separation ability. So what are the functional differences between the diffused state and the condensed state? So one of the things that DDX5 does best is utilize ATP. So we use this as a surrogate to measure its activity. So what we've done is we have a pre-steady state droplets. We had to separate these droplets because they form, once you get a purified solution, if the protein concentration is right, in a few hours, you get this steady state. So we spun the droplets down, and then we divided the solution into 2. The first one was very fast measured for ATPase activity, and the other fraction was left to mature, to reach pre-steady state, and was basically a mixture of droplets and diffuse form. So we measured ATPase activity in both of these fractions, and we're able to demonstrate the fact that it seemed that the droplets are actually more active than the diffused state. And if you repeated the same experiment with the delta purity, we saw that now the activity is still there. So the activity of DDX5 without the delta purity is still there, and it's quite comparable to the wild type, to the full length, but the differences between the droplet and the diffuse state is now abolished. So I was able to talk to you earlier about a prion-like prediction, and we wanted to see whether truly this prion-like domain also could be responsible for prion-like propagation. So to test this, we use a very elegant assay developed by the Jarosz lab. And what we do here is actually quite simple and elegant. When you express the GFP tagged version of DDX5 in a basal level. It does not aggregate. You can see it localized very nicely to the nucleus of yeast. Everything is done here in yeast. And as you can see, the red tag version is not expressed. If we boost the expression only of the red tagged version, it starts to aggregate because we increased its concentration. What we see is that immediately the GFP tag version starts to aggregate itself as well. So there could be several possibilities why. The two most obvious ones is maybe by overexpressing the red tagged version, we actually stressed the cell and things didn't fold well. The other possibility is that maybe there is a seeding behavior, that the red tagged version is now seeding the green one. So to try to distinguish between the both, we reduced the expression now of the red version, diluted the culture, and now let them propagate for many generations. But what we saw is that once this Prion-Like switch actually happened, it's now stable. The green puncta, the green aggregates are now stable for many generations as long as the basal expression of the GFP tagged version is actually enough to fuel this reaction. So we tested many controls. We overexpressed just mRuby. We overexpressed polyQ tagged version with mRuby, and neither of these options actually induced aggregation of the DDX5 GFP tagged version. And we also tried to delete the PRD domain, and this actually was proven to be very, very important for this behavior. This is an example. If we perform this experiment with full-length protein, you can see that the green immediately induces aggregation in the red. But if we now delete the Prion-Like domain from the red version, it's by itself can still aggregate, but now it lost its ability to transmit its conformation to the green tagged version, basically uncoupling aggregation from Prion-Like behavior. So what I've shown you in the second part was that we identified DDX5 as a protein that has an increased aggregation propensity with older age in the vertebrate brain. We were able to show that there is a balance between a diffused and a droplet state, also in vitro, and potentially also aggregation or maturing of these droplets to full-blown aggregates. And we propose that maybe with aging, this steady state, this balance is actually disrupted. And once you start accumulating these DDX5 aggregates, they can potentially start fueling themselves and accumulate. We were also able to show that from our preliminary data that the condensed version of DDX5 is potentially more active functionally, which proposes one of an intriguing possibility that maybe there is a reason why these droplets form in vivo specifically for DDX5. And our next steps is try to isolate these aggregates and inject them into the brain and see if the fish and see if they can actually propagate in the brain of the fish. We can assess behavior. This is an automatic behavioral chamber that we can use to assess behavior. We can use transgenics to view the brain through the skull of the fish, or take the brain out and explore it more in depth. So I mentioned the people involved in this project in one of the previous slides, but I'd like to thank my newly established fantastic lab at the Hebrew U, our funding sources, and thank you very much for having me.
Itamar, thank you very much. Nice work from your new lab. So if I could have control of the screen. So you'll have to stop sharing, Itamar.
So yeah, sorry. Control. Yeah, I give it back.
Thanks. And I'm going to talk today about nuclear condensates and gene regulation, cancer, and drug partitioning. And then I'll lead our discussion at the end of this. So Roger asked us some questions about specificity and function, and I thought I would just summarize very briefly what I've learned from the leaders in this field. And I've noted them at the bottom of this slide. I know there are others, but I just wanted to give a shout out to these specific people because of their influence on me.
Rick,
and of course,
you're sharing the wrong screen. I think we're seeing your presenter's view. Slides. Sorry.
Thank you.
Yeah, great. Thank you.
So, you know, what we're learning is there's compartmentalization and concentration of components. We're learning that there are diverse modifications that can participate in the partitioning selectively of molecules into condensates. This area allows us to move beyond thermodynamic modeling of equilibrium conditions. And I think we're uncovering, and you've heard about them, novel disease mechanisms and therapies that are inspired by understanding condensate physicochemistry. And I'll talk about this last subject. First, I'm going to talk a bit about condensates and gene regulation, how those are dysregulated in cancer, and then I'm going to focus most of my talk on partitioning of cancer drugs. And I've noted my conflicts at the bottom of the slide. So what we have learned, and Phil Sharp introduced you this, is that in the world of transcription, we think, at least at the largest super enhancer driven genes, there are phase separated condensates that occur. These are very dynamic. As you heard from Phil's talk, Ibrahim Sisi has taught us that the larger the condensate, the greater the transcriptional output. These condensates have lifetimes that can be measured in seconds to minutes, and the larger condensates are more long-lived. And what we learned is that transcription factor activation domains are playing an important role together with the DNA they're binding, which crowds them, and together with cofactors, they will undergo what we believe to be a liquid-liquid phase separation and create condensates in vitro and in vivo. And one of the reasons we're convinced this is the case that all the data we know of in transcription factor activation domains, their ability to activate in vivo also affect their ability to form these condensates. And moreover, ligands for nuclear hormone receptors that are essential in vivo for example, estrogen receptor function, also are essential for the phase separation that we see in vitro. And so these models that have emerged from multiple labs, Ibrahim Sisi's lab, my lab, and others, help us explain how activation function, hundreds of different transcription factors can interact with a single type of mediator complex. Often these 200 to 300 nanometer diameter condensates have hundreds of molecules, each of mediator and Pol2. And we think this can help explain why multiple labs have seen that enhancers can activate genes without coming closer than about 200 nanometers. All of the features we know that are important for the regulation of gene expression are altered in cancer. So we see current events in enhancers in what we think of as the master transcription factors. These are also classically termed lineage regulators. They're the factors that are expressed at unusually high levels and really drive cell state, and factors that can actually be used to reprogram or transdifferentiate cells. These are misexpressed in tumor cells. Oncogenic signaling is recurrent changes in epigenetic behavior of the cell are recurrent features. And non-coding RNA, although poorly understood in this context, is generally altered in tumor cells. And so what we've come to understand is that tumor cells are acquiring, they're evolving large super enhancers at their driver oncogenes. And the classic view of enhancer hijacking and transcription factor dysregulation is in fact forming these large condensates at the driver oncogenes. We've come to understand now that the behavior of a small indel that can put a single binding site for one molecule of highly expressed transcription factor in a tumor cell can actually take that portion of the genome from close to a super enhancer driving an oncogene. And that kind of extraordinary cooperativity we can only explain now in the context of overcoming the threshold separation process. We now understand that oncogenic signaling is delivered preferentially to these super enhancers. Most of those terminal factors in the signaling pathways have these oligomeric repeats or IDRs that are participating in fusion with the super enhancers at these driver oncogenes. And so they're effectively physically linked with the genes they're going to regulate. But one thing that we failed to understand, at least so far, is why super enhancers are exquisitely sensitive to drugs that target general transcriptional regulators. And we saw this first with Jay Bradner with his famous JQ1 tool compound that binds to the bet-bone domain protein BRD4. We've seen it with CDK7 inhibitors. These inhibitors that should affect global transcription, instead in these tumor cells have these very selective activities. And although we've come up with another number of models to explain this, none of them have really been terribly satisfactory. But I'm going to give you a model that might explain this. And it comes from thinking about how drugs exert their pharmacological activity. So what you see on the left are one or two criteria in pharmacology, pharmacodynamics. This is how a drug affects its target. And what you're looking at here is this beautiful structure of a portion of the estrogen receptor where you can have binding of either estrogen or an anti-estrogen compound. And anti-estrogen compounds are typically used in breast cancer and in cancers driven by androgens. There's also a field called pharmacokinetics, and that is really regarded as what you yourself do to drugs. And that focuses on the absorption, distribution, metabolism, and excretion of drugs. But we have assumed in the distribution part of this equation that once in cells, drugs typically access most compartments. They're free through most compartments. We've been examining whether nuclear condensates occur both in normal and malignant tissues in individuals. So far, we've only examined these in the context of tissue culture cells. And so it was reassuring to see that the scaffolding proteins for the various kinds of nuclear condensates that we've been studying are all in these puncta that have observed by others in tissue culture cells and by us. As you can take what you believe to be scaffolding proteins for condensates, and you can reconstitute them with purified proteins in vitro. And I'm just showing you that that can happen for this set of transcriptional splicing, heterochromatin, and nucleolar proteins. So what we wanted to know is whether small molecule dyes, small molecules that are fluorescent but have about the size of a therapeutic drug, will freely diffuse through these in vitro formed protein condensates. And this data just shows you that in fact they do. They are not especially enriched or excluded from these in vitro condensates, as shown here for fluorescein, and similar results have been obtained with Huxt and with 4-kilodalton labeled dextrans. In contrast, however, anti-cancer drugs such as mitoxantrone can be concentrated selectively in certain condensates. And it's been known for some time that mitoxantrone, a drug that is used in some forms of cancer, will concentrate in the nucleoli, where, as you probably know, Fib1 and NPM1 play important roles in phase separation. And what we think now, by looking at these in vitro form droplets, that at least part of the reason mitoxantrone is able to do that is not because its target DNA is there, it's a DNA intercalator, but rather because of physical chemical properties of the condensate itself. And what you see here is that it will partition into MED1, Fib1, and NPM1 droplets. You don't see it in the top of this diagram. You don't see the transcriptional condensates that are formed by MED1 because they are so small relative to those formed by these nuclear proteins. We were curious about other anti-neoplastic drugs, and the most widely used anti-cancer drug are the platin family. And so we looked at cisplatin. And what you can see here is that cisplatin partitions selectively into these MED1 droplets. And in work we did with Tony Hyman's lab, we were able to determine that the partitioning is such that cisplatin can concentrate about 800-fold in these MED1 droplets over the surrounding bulk. Cisplatin, and in these MED1 droplets, you see highly mobile proteins and drug. The drug is a bit more mobile than the MED1 protein, but that mobility suggested to us that there are weak transient interactions, and of course, those could involve in principle salt bridges or pi-pi or cation pi interactions. We got together with a chemist, Young-Tae Chang in South Korea, to examine a library that he'd created with a fluorescent Boddipi moiety. This moiety allows you to add a variable R group at 2 sites, and by creating a diverse chemical library, we could then ask whether or not we could identify chemical functionalities or features of these chemistries that might be responsible for enrichment. And when we did that, we found that aromatic rings were selectively partitioning into MED1 condensates, as you can see on the left. And that suggests that there are pi-pi or pi-cation interactions. And the bottom of the list of partition were these non-aromatic ring containing compounds, where, as you can see here, there's no enrichment within the MED1 condensate. So that led us to ask whether or not there were specific amino acids within MED1 that are important for this partitioning behavior. And MED1 phase separation in vitro depends on a serine patch that exists in that protein. But we were curious about whether or not basic amino acids or aromatic amino acids within MED1 might be responsible for this drug partitioning property. And what we found was the basic amino acid changes had no impact on this partitioning, but the aromatic amino acids, when substituted, caused the loss of this partitioning by our top Boddipi probes, and in fact by cisplatin. So we could separate the condensate promoting abilities of the serine patch in MED1 from the cisplatin concentrating properties of MED1, which are conferred by these aromatic amino acids. What we were curious about is whether or not the platinated DNA occurs preferentially in these MED1 condensates. So we wondered whether or not MED1 condensates with DNA we're making HP1 alpha condensates with DNA. It would give us an in vitro assay of cisplatin partitioning and allow us to ask whether or not the reaction was a bit faster with MED1 because of this partitioning behavior. And this slide shows you that that is true. As you look across a concentration course of cisplatin, what you see is that the platinated DNA is much more platinated over that time course if it has been incorporated into a MED1 condensate than if it's an HP1 alpha condensate. But what about in living cells? So we did this assay in a variety of tumor cells, and what you can see from the slide, first of all, in the left, is if we take an antibody against platinated DNA, so it's not recognizing cisplatin, it's recognizing the product of the platination reaction, then do ChIP-seq with that antibody, what we see is that super enhancers are especially enriched in platinated DNA. And moreover, if we do this by microscopy and we look at nuclear condensates, and here we've examined 2 sets of cells each for MED1 containing condensates, HP1 alpha containing condensates, and Fib1 containing condensates. And what you can see by imaging for platinated DNA again with this specific antibody is that only with the MED1 containing transcriptional condensates do we see an overlap of the platinated DNA with the MED1 condensate. Now, if we allow that reaction to go for longer periods of time, we see something that you might expect, which is that we are causing the loss of MED1 condensates selectively. So what you're seeing here is after 12 hours of platinum treatment, these tumor cells are losing the MED1 condensates, but they're not having any obvious effect on the HP1 alpha condensates. And there's a time-dependent effect that we see with these MED1 puncta. So this partitioning property that I've described for cisplatin also occurs with many of the other drugs that are used in the context of cancer. So this FLT-X1 drug is a tamoxifen analog. It's a fluorescent version of tamoxifen that actually acts clinically just like tamoxifen. And what you see is that it partitions nicely into MED1. GQ1, this drug that interacts with BRD4 bromodomains, is partitioning into MED1 and BRD4 condensates. In this case, the BRD4 molecule lacks that bromodomain that is normally doubly affinity interaction. So JQ1's partitioning here by properties that are independent of its target. And THC1 is a CDK7 tool compound. Like these other compounds, it's selectively partitioning into MED1. And as you probably know, CDK7 resides within these transcriptional condensates in living cells. So these drugs that are general inhibitors of ER or these cofactors, BRD4 and CDK7, have very selective effects in cells. And now we imagine that the reason we're seeing that selectivity is at least in part due to the concentration of these drugs in super enhancer condensates, which can occur, at least with cisplatin, where we've measured it up to 600-fold greater concentrations than in other sites in the cell. So I'll just close by saying, I think this is a very exciting area because it brings us to trying to understand the physicochemical codes for interactions. You've heard a lot of exciting work on the features of amino acid side chains and of RNA molecules that play roles in partitioning. We'd like to now know in greater detail what kinds of chemical functionalities are playing these roles. with the goal that we might ultimately be able to target drugs to specific portions of the genome. And I'll close here just by acknowledging the extraordinary work of Anne Boyja and Isaac Klein and their colleagues, and my collaborators, Arup Chakraborty, Ibrahim Sisi, Nathaniel Gray, Tony Hyman, Rudolf Jaenisch, and Phil Sharp. So what I'd like to do now is move to the discussion part of this wonderful meeting. And I, at the beginning of this meeting, Roger Kornberg asked us two specific questions. He said he would like to know more about what is involved in specificity. Biochemists, Roger, need to understand specificity so that we can better understand his second topic. He wanted to know how condensate behaviors impact cellular functions. So part of discussion by telling you, first of all, that Julie Foreman-Kay and Amy Gladfelter, Tony Hyman, Rudolf Jaenisch have agreed to be present to answer questions. In addition, any of the speakers from today should be available. But I'll just begin by asking Amy Gladfelter a question about specificity. And Amy, if you're there, could you tell us how you think about specificity in condensates?
Hi, yes, I'm here. Thanks for the great talk too. This has been such a fascinating meeting. So I think we touched on specificity in multiple times during the meeting. However, I think this is probably still one of the hardest puzzles for both the protein and the RNA. And I think RNA still remains a lot of mystery. I think the second challenge of activity we're making more progress on in terms of RNA biology. I think that was really seen here, today, looking at DDX5 chaperone activity, looking at hearing from Julie yesterday on deadenylation activity. But the real question mark is how are these enzymes, which may be modulating have their activity modulated, how are they finding specific targets, and what are the specific targets? And so I think the framework of specificity, we're beginning to see it. We see it from Tanja at the protein level with the stickers and spacers framework. Our work has looked at how RNA houses stickers, but really the complexity and the heterogeneity within a cell. So I think we don't even begin to understand sort of the heterogeneity even within a particular class of condensate within the cell. So I think specificity is still a problem for many, many people to tackle for a long time.
And Julie,
So yeah, thank you, and thank you, Amy. I hope the noise. Yikes! There's some renovation in my house, but the point that I wanted to make is specificity may be there. We can get some insights into that by thinking about condensates as solvents, and instead of trying to understand individual, this group binds to this group, we can think of, for instance, when you have an extraction with organic solvents, you pull some components out and you leave others. So if we think about the combination of proteins and RNA in these dynamic interactions, creating very unique solvents. So we have individual condensates that behave like unique solvents that are doing their own mini extractions of the cytoplasm or the nucleoplasm. And that's what I think we need, that could be helpful in understanding that. So the dynamic interactions of these chemical groups are what create the specific properties of the solvents, and those will change depending on what's in them, and the surfaces that they're built on, and the DNA or RNA, and the RNA structure will make that solvent environment change. So that's not that we've solved it by what I just said, but I think that that's an insight that can be really valuable moving forward.
Thank you, Julie. Tony, could you add a little bit to that?
Yeah, I mean, I think, Amy and Julie stated that very well. I think one thing I'd like to say is that the concept specificity probably needs to be expanded a bit in the condensate field, because coming from a molecular background, specificity is a very, very specific meaning about how you select one molecular interaction from another, and defined by binding constants. But in the condensate field is also compartment identity, where the compartment forms, and those are other issues of specificity that are equally important. I mean, one way I like to think about it as well is that the specificity can be seen as a term I've been using recently on molecular communities, that a lot of molecules like to phase separate and be with each other. As an example, we did some experiments with the fuzz family, and if you make 30 of those proteins in vitro of the different family members, 20 of them will all partition and like to be with each other, and then one has a coil and it now demixes from all the others. But once you have a community of proteins that say 30 or 40 like to be with each other, then a lot of the biochemistry we've been talking about can happen in a sort of smaller context. In other words, a protein, instead of having to distinguish between 10,000 other proteins, maybe has to distinguish between 50 other proteins in the context of the community. And that would allow biochemical reactions to be more specific, as an example, in this local community. And once you think a little bit about specificity in that sense, it's not just binding constants that will be important. It'll be things like shape or degree of disorder. Those are things which will make proteins want to be together in one place rather than another, rather than the more conventional concepts of stereotypic interactions that comes from all the molecular biology we studied. So that's, I guess, my summary there, is that the interesting thing is to understand how the traditional interactions defined by binding constants, specificity we think of, we all learnt about from high school at this point probably, sort of meshes together with these concepts of specificity which define compartment identity and compartment position.
Tanya, if you're there, you talked about spacers and stickers and the various features that are contributing to condensates. Could you add your views on how specificity might be acquired?
Yeah, thanks, Rick. I think Julie brought up a very good point in terms of thinking about the condensate more as a solvent. I would say that, of course, these solvent properties are still encoded in the sequence of the constituents, and perhaps by how ions, small molecules are recruited to these as well. I think there is additional complexity in the cell where we're, of course, not in equilibrium. So there could be kinetically encoded or specificity as a consequence of kinetics, of things moving more slowly into one condensates than others. So I think this is another component that we need to think about.
And for people who have questions, please type them into the chat. You're welcome to add to this discussion. But while you're doing that, I'm going to turn to Rudolf Jaenisch and just ask him to comment on condensate behaviors and how they impact cellular function. You described, Rudolf, how you think this might happen in the context of MECP2. Would you like to add a little bit to this discussion about function, cellular function? Yes.
So what I said was sort of a puzzle that MECP2 deficiency really activates a number of genes, 500 or so. So it's like a repressor of those genes, but it also really inactivates thousands of genes. So it's probably like a reason to think that the repression is a direct effect. The activation is probably indirect. It might be, and we don't know what's happening, it might be a redistribution of other factors from other condensates, let's say transcription factors, which then inhibit and these genes. So this is really a puzzle. What we know is really from the study of these mutations in the Rett syndrome is that the disease-causing mutations correlate with condensate formation. This does not say it's causative, but it's a correlation. So I think what we really need to do is to see whether it's correlative, to affect the condensate formation by whatever, maybe by small molecules like Rick described, or other means. But we don't know exactly what we have to do. Do we have to enhance condensate formation? Do we have to inhibit other condensates maybe, or enhance them? We don't know. So I think this is an outstanding question at this point to really prove that there is a causal relationship between changing the condensate formation in this disease and the phenotype.
Thanks, Rudolf. There's a question, Amy, from Hayoung Yu. He wants to know, how does phase transition of the nucleocapsid protein distinguish viral RNA from the host RNA?
Well, that's a great question, and I wish I definitively knew the answer, but I can tell you how we're thinking of it, which is that there are, we think, very specific structural elements in the genome that are essentially enabling nucleocapsid to bind and are basically multivalent binding sites. And we think these are actually positioning the nucleocapsid in a particular way that's promoting phase separation. And we predict that this is not present in the host RNA, that these specific binding sites. But, you know, that's work we're really intensively doing right now. But that's my best answer at the moment. Thanks for the question.
May I add a little bit? I think if you can just express those specific structures, maybe those can impair viral packaging.
Yeah, we're doing experiments right now, honestly, along those lines. So I hope we can get to the bottom of it. I just don't have clarity yet.
And Amy, another question from Meta is, how do RNA structure and flexibility generally compare to protein structure and flexibility in this context?
Oh, that is an excellent question. And I think it all depends on the context. So the persistence lengths are really going to depend on the degree of structure within a given RNA, and similarly to a protein. And I think I would like to say that particularly for RNA, I don't think we have great empirical estimates of this on real long RNAs that are, you know, the native RNAs in a cell. I think these are really early days in understanding the mechanical properties of RNA and how they contribute.
Thank you. This is so exciting.
Thank you, Amy. I think this question's for Daniel Harries. Taking the analogy of phase from molecules to solvent, could we analogize the conformational preferences of IDRs to solvent structuring, like the preferences of liquid water?
So definitely, I think structure of solution is definitely at the heart of a lot of this, and the analogy can be to aqueous solutions, but also to others that we know, solutions that contain polymers and so on, and I think one thing that I am taking from this meeting and from this community is that there is really a huge importance to local variations in concentration. So it's coming away from these classical models of phase separation where there is an all or none kind of phase, and the local concentrations and variations become really important in driving forces, additional forces, through solution to make these condensates or aggregates what they are. So definitely yes, but I think there is another layer of complexity that is coming from local variation in concentrations.
Thank you, Daniel. And Tanja, there's a question for you. For the NMR experiments, how do you keep proteins in a monomer state? And he wants to know in phase diagrams, how are concentration and temperature related?
Mm-hmm. Well, so it turns out these the phase transitions of these IDRs are very highly cooperative. So basically below the saturation concentrations, they are monomeric. We've actually done some careful experiments to look at whether we see any sign of any oligomers or clustering. And, you know, if you look very carefully, there is a very tiny amount right at the saturation concentration, but basically they're monomeric. And so you just need to use conditions that are on the left side of the coexistence regime, on the dilute side of the coexistence regime. And in the protein that we've studied, this is a UCST, upper critical solution temperature system. So at increasing temperature, there is a lower driving force for phase separation. Of course, there are other systems in which this is reversed, where you can have an LCST type system where you get phase separation when you increase the temperature.
Thank you. From Ado, Tony Hyman, the question is, molecular crowding plays an important role in cells for phase separation. Many options are available for the in vitro experiments, for example, dextran, PEG, FICL. How do we the use of these? And I think others may want to weigh in on this. I know Tom has written about it, but Tony, if you're there, maybe you could start.
Yes, I'm not totally sure what I mean by this term logically, but you know, different types of crowding agents have different effects depending on how they're acting. So sometimes you just have a water crowding effect, and then basically all of them will work. But other cases, you'll have more specific, such as effects of, say, length of dextran, and then you'll want to adjust the length of the dextran to find the best crowding conditions. I mean, the theory behind them is not that well developed for the molecules that we use. So generally, you just have to take them all and experiment for which one is best in your particular case.
Yeah.
Tanja, do you want to add some more to that?
I think Daniel gave a great talk on this, right? So perhaps he wants to make some additional comments.
Daniel.
Right. So I think what we should remember is that any of these crowding agents that are used do more than just crowd. So they also interact with the molecules. I think it's now well acknowledged that it's not going to act just through limiting volume. And so it's all these molecules will do more than that. So polyethylene glycol on occasion can denature proteins, not only stabilize them. And this goes to these so-called quinary interactions that are on top of any kind of volume effects. So all of that has to be taken into account. There are some experiments that show what kind of interactions these different macromolecules have with each other, and that information can definitely be used to decide what should be the choice of environment for these macromolecules.
But I think it's a sort of follow-up to that. Danny put it very nicely. It's just that, you know, if you take two completely different crowders and they give you the same effect, you can be sort of happy that you're probably seeing a crowding effect rather than some specific interaction. That's why you don't really want to just rely on one particular crowder when you start your experiments.
Just a personal observation, you know, many of us are effectively studying physicochemistry, but from my personal point of view, doing that with a license. And you know, I know this is a challenge to everyone who works in this area to actually have command of the underlying physics and the underlying chemistry in what is happening in our assays. So I think that's going to be a challenge going forward. We're working at the cutting edge in some cases of these sciences. There's a question to Itamar Harel. You showed that PRD alone wasn't enough to propagate the prion-like behavior of DDX5 protein. Have you tried the same experiment, but only with deletion of PRD, and analyze if DDX5 has prion-like behavior by forming condensates alone?
So actually, this is the experiment that we've done. We've done the deletion of PRD, and that version did not appear to be prion-like or propagate in a prion-like manner. I did not show you our experiments with overexpression of only the PRD, and this doesn't even aggregate. So we see tiny puncta, but they don't aggregate at all. So it could be that because, you know, the PRD itself is a small enough piece, and when you fuse it to GFP has a solubilizing aspect. But what I've shown you in the presentation is a deletion of PRD from DDX5, and that aggregates, but cannot propagate in a prion-like manner.
Thanks, Itamar.
Just to add something about specificity, with prions, you see extreme specificity. For example, we did an experiment in which we co-expressed the human DDX5 and the killifish DDX5. And even though either of them can propagate amongst themselves, there is a species barrier between them. And this is just something that to consider. I don't know yet. We're still thinking about the relations between prion-like propagation of the aggregates versus the droplets and the phase separation. But it's just fascinating to me that the specificity also occurs when these proteins also aggregate completely and not only as a droplet.
Thanks. And I've also been impressed with the extent to which classical post-translational modifications of proteins, and I imagine RNA, will create selective differences in partitioning of a biomolecule into one condensate versus the other. So I think in the future, it's going to be very interesting to see how many examples of what we imagine are classic regulatory mechanisms in molecular biology, where phosphorylation or acetylation, for example, can modify the behavior of a protein, is also modifying the partitioning behavior of that protein and its friend in a shared function. I wonder if others in the meeting have this experience. I think, Julie, you may have been talking about this to some extent.
Yeah, so, you know, one thing that was interesting in the beginning when we were looking at post-translational modification of these low complexity IDRs, a lot of people wanted to know which position was important. So the specificity of phosphorylation dependent effects, for instance, in enzymes, they often want to know which site. And it turns out that it's more of a polymer property and more of a solvent property that you have multi-site phosphorylation that changes the physical chemistry of the chain. And it depends, in not completely in a sequence independent way, but even more generally on how many sites are phosphorylated than on the individual positions. And we've seen this with arginine methylation, and now looking at O-glucnac, similar effects where you're really changing the physical chemistry of the chain. And those changes cause specific solvent-like differences as opposed to needing specific positions to be modified.
I'll just add a counter model to that, which I think is just important, that I think there's going to be a lot of different ways that PTMs may work here. And that is, in the We3 system, we have seen residue-specific phosphorylation sites that are actually separation of function, basically, that are deterministic. And those have been mostly mapped in vivo and analyzed in vivo. And so, we don't know the basis for it, but I think it'll be really interesting to think about different scales of PTMs. I think there will be polymer level scales, but then there may actually be more specific molecular scale effects.
Yeah, well, for certain, if you make an SH2 binding site by phosphorylating a specific tyrosine, you're going to be able to have like modular binding domain motif type driven phase separation. So there's also going to be those kinds of effects. And I think those are also very important. So yeah, Amy, you're right. There's a whole range of ways that PTMs are going to affect this.
In my own field, there is a portion of the large subunit of RNA polymerase II that is a heptapeptide repeat that is repeated 52 times in mammals and is the size of an average protein. And it's a giant IDR that helps polymerase apparently partition properly into condensates that are involved in transcription. Patrick, a very, very talented crystallographer who has really described in great detail the atomic structure of the portions of RNA polymerase II and its cofactors, emphasizes how they have to cut off all of the components of these transcriptional apparatus, including that IDR, in order to get high quality crystal structures and avoid untoward behaviors of purified proteins. And he cites the need for that IDR to be hyperphosphorylated to facilitate a transition from initiation to elongation. So I think there are examples where PTMs, that's a rather extraordinary example where there's a portion of a biomolecule comes in with this huge domain, relatively unphosphorylated, becomes hyperphosphorylated in this transition. And that's quite an important function. If you mutate the kinases that are responsible for that transition, you don't get it. I just thought, maybe we just have a couple of minutes left. There are a couple more questions, but I thought maybe I would just throw out the point that this field is so young and exciting, and the Hebrew University and this institute have provided us with a forum to have this remarkable discussion. I think we should thank Roger Kornberg, Nissim Ben-Vinisti, Eran Bashar, and all the participants for really what I think is an extraordinary meeting. So thank you all.
Thank you, Rick, for really, for all the lecturers, for fantastic talks, and for this exciting discussion. I would like to share with you that we were using this platform of this conference with the best lecturers in this field to have a very short course for graduate students. And the homework of the graduate students is to choose one lecture and write a very short research proposal based on the lecture that they like the most. So beware, you are going to have competitors from our graduate students. And really thank you all for really very exciting meeting. Eran, myself, and Roger was planning to bring you all to Israel. It didn't work this time. Thank you for doing that virtually. And we hope to see you all when this pandemic will be over. Eran,
Yes.
do you want to say some words?
Yes, I first, of course, I join Nissim in thanking all the speakers yesterday and today. It was really a fantastic meeting and a learning experience definitely for us. And I'd like to thank also the organizer behind the scenes, Smadar Bergman, Ilana Gutkin, and of course Anat Yagil, without whom this would not have happened. And of course, Professor Yitzhak Chen, who took up our initiative and is the director of the advanced school. And of course, Roger and Nissim, chairs, speakers. It's really been a fantastic event. So thank you very much. And we look forward to seeing you all in person, hopefully soon. Thank you, guys.
Thank you. That was great. I enjoyed it very much, and look forward to seeing everyone in Israel when the difficult times are over.
Yes. There are actually a few unanswered questions. So if you happen to be one of those people who the question is for you, you can stay another minute and try to answer them in the chat. I see a question for Reut, for Elon. So try to see if there's a question for you.
I just answered on chat because I saw time is running out.
Okay, great. Thank you. All right. So, bye-bye.